Embryonic development will give us our next division of derived characters on our tree, and this actually looks at the process of gastrulation. If you remember gastrulation, that was the formation of a digestive tract, and it involved the folding in of our blastula. So our blastula was a hollow ball of cells, and if we fold that in, we get a gastrula, which is going to be the formation of our early digestive tract. Now, if you remember, when we focused on this sort of development before, we looked at this development of this early digestive cavity, and really, this organism could be anything at this point, right? So what we need to think about is how we go and then develop into a further body plan from this. What we focused on when we started to think about these organisms and this development was the blastopore, that folding in spot, and whether or not that became mouth or anus. And we'll talk about that more later. But this folding in gives us these body layers, and that's critical for us in the development of different organ systems and layers to our body. We have talked before about this. I have this here to just kind of remind you. We can think about changes in embryo structures and into adult phases. We go through something known as metamorphosis, which is change. And typically we look at that larva, this ball of cells we were talking about, going through pretty significant change to make it look like you, right? Or any other animal. Remember, when we talk development, all of these things happen because of these homeotic genes, or these master control genes. And what we're hoping that they are going to do for us is help us form these tissues, germ layers. And when we look at these germ layers, we have two possibilities. We have animals that have 2 germ layers and some that have 3. Well, if you have 2 germ layers, you are a diploblast. If you have 3 germ layers, you're a triploblast. Remember, di is 2, tri is 3. Why is this important? Well, when we look at this, our diploblasts only have an ectoderm and an endoderm. If you remember back to development, ectoderm forms the outer layers, so our skin and our nervous tissue. The endoderm forms our critical organs on the inside. The mesoderm actually is going to give us muscle structure and bones. So if you have an organism that is lacking a mesoderm, like you do with our diploblasts, you can think about them as lacking some of these advanced muscular and bone structures. So here are our protostome and deuterostome. You remember this blastopore is forming and folding in to form those germ layers, and as it folds in, whether or not that blastopore becomes the mouth or the anus will be important for us when we divide out our tree even further. And we've talked about this before, this folding in that occurs, and whether or not that first folding in of the blastopore is actually the mouth or the anus determines what you are going to do as far as further development, and eventually how we're going to split you out onto our cladogram. Now, when we look at this development, we do look at this folding in of these embryonic layers, this folding in of this blastopore, providing space and structure for internal organs to develop. And we develop something known as a coelom. We've talked about this before as being a true cavity. So this coelom, or cavity, is an open space. When we look at a true coelom, we can look at it being lined by tissues. This is a very advanced structure. A pseudocoelom, remember pseudo is fake or false. So this is a body cavity that's not going to be lined. In particular, it's not going to be lined with mesoderm. So when we look at these pseudocoelomates, a group of organisms with this false body cavity, we'll look at them as only having 2 body layers or being diploblasts. you need to connect these terms together. So if we're talking about an organism that doesn't have mesoderm, you need to also think about the fact that it's a diploblast, and you need to think about the fact that it's not going to have a true body cavity, and you need to think about the fact that that will then make this organism less complex in the long run. So when we look at these body cavity divisions, we actually start to divide out more of our phyla. This is a funny word for you. Platyhelminthes refers to our flatworms. Okay? So when we look at our flatworms, this is a beautiful one known as a nudibranch. Very common in the oceans. When we look at these, we are looking for ways in which these organisms can be structured and divided out on our cladogram. That's the whole idea, right? We're building that tree of life, but we also have to think about what it means as far as their complexity. So a flatworm, like I just showed you, it has ectoderm, mesoderm, and endoderm, but it's not going to have a cavity. You see? There's no coelom here. So it has no internal cavity. So as soon as I tell you that, you need to think, okay, that's no space for internal organs, right? But if we look at our roundworm, these are also known as nematodes. We often look at them as being parasitic. They start to get this pseudocoelom that I mentioned to you. So there's a space, but it's not going to be lined completely, meaning that they can have some organs starting to form in this space, but they're not going to have the same organ structures as if you look down here at the earthworm, which falls into our annelid category. And you may not think about an earthworm as being very complex, but it has a heart and a gut and all of these support organs associated with it filling this cavity or coelom, this inner space that allows higher level development. So now that we've broken out our sponges as our out group, we can break out our Cnidarians as being the only ones with radial symmetry. Then we can start breaking out our flatworms, nematodes, and annelids based on how they are structured with their internal cavity. Annelids down to chordates are all going to be triploblasts, and they're going to be what we call coelomates.